So let's start the discussion for week 4. So here goes the first question. So lectins are what lectins are? These are either carbohydrate binding carbohydrates, protein binding carbohydrates, carbohydrate binding proteins, or
Protein,
lipid binding.
protein binding carbohydrates.
Okay, any more answers? So yes, these are protein binding carbohydrates, carbohydrate binding proteins. Sorry. So these are basically membrane proteins that basically binds to the. This is a typical structure of lectin that you all are seeing in this particular slide. So, like in this structure, we can see that these are membrane-bound proteins. Okay, so this is the cell membrane that we see, and these have several domains. This is the transmembrane domain that helps anchoring these proteins to the membrane. Okay, and this is the cytoplasmic domain that basically helps in signaling. So if they attach to any particular carbohydrate moiety, so they have to transmit signal to the inside cell so that they can further process different mechanisms inside the cell. So cytoplasmic domain helps in signaling of these molecules. And then comes the repeats. These repeats are basically helps in clustering of different lectin molecules. So what happens whenever there is some signaling going on, or specifically when there are some reactions going on? So these repeats cluster themselves with one another and form this kind of mesh kind of structure. So this helps in a particular robust signaling and binding to the moiety present on the uh, other side. Okay, and then we have the last part, that is the important part, that is carbohydrate binding domain, or carbohydrate recognition domain. So this particular domain recognizes specific kind of carbohydrate. Okay, so for example, there are lectins specific for mannose. So this will be specific only for mannose. So it depends upon type of cell it is present and the signaling it is going to perform. So for example, if it's a dendritic cell, so what will happen? This carbohydrate recognition domain will have a moiety recognizing all the antigen molecules that are present either on the infected cell or different pathogens that are being that infect our body cells. Okay, so this is the typical structure of a lectin molecule that we see in our cells. So anybody has any doubt? If not, then we'll move on to the next question. So here are two statements given. We have to select which one is correct and which one is false. So the first statement is lectins can bind to glycolipids, and second one is lectins cannot bind to glycoproteins. So can anybody deduce which statement is correct?
Sir, option C.
Okay. Any more answers?
Sir, option D.
Option B. Okay. Any more answers?
Option B.
Okay.
Option
So
D.
the correct answer is option C, as you all can see, because like uh, if we consider a lectin molecule, for example, here, so this is the carbohydrate binding domain. So the carbohydrate that is binding can be a glycoprotein or a glycolipid or any molecule of the cell that is bound to the carbohydrates. Okay, so lectins will recognize anything that is bound to the carbohydrates, specific carbohydrates. Okay, so yes, so lectins can bind to glycolipids and they will also bind to glycoproteins also. So the option C, that is statement one is correct, but statement D is false. So option C is correct. Okay. So anyone has any questions? If not, then we'll move on to the next question. So hemagglutinin is a dash that binds to RBCs and causes dash. So can anybody fill in the blanks?
Option D, sir.
Option D. Okay. Any more answers?
No, option C.
Okay. Any more answers? So the correct answer is option D. So hemagglutinin, that is like for example, it's present on influenza virus. Okay, that's why these are named as in the series of H and N's. So H stands for hemagglutinin and N stands for neuroamylase. So what is this hemagglutinin? It's a lectin protein only. Okay, that binds specifically to carbohydrate moiety on RBCs. As you all might know, that the antigens present on RBCs that determine our blood group are basically carbohydrates only. Okay, so they are different kinds of carbohydrates modified in different versions. So that make our blood groups. So lectins, especially hemagglutinin, recognize these carbohydrate moieties only, and these causes the agglutination reaction. Okay, so this agglutination can be used for vaccine development and other things also. And this is the major cycle of infection also inside the body. So this is how the hemagglutinin works. Okay, so it's a glycoprotein or a lectin molecule that is carbohydrate binding protein, and that binds to the carbohydrate moiety of the RBCs. Okay, so we'll move on to the next question. So rods and cones are specific type of cell receptors. What are these and what different rod sense and cone sense? So can anybody guess?
D, sir.
Sorry, can you repeat?
Option D.
Okay. Any more answers?
So option D.
Yes, the correct answer is option D only. So to explain, so where the rods and cones are present, these are the major integral part of retina, and these are collectively called as photoreceptors. Okay, so this is the back of the eye, and in the back of the eye we have the retina structure, and after that we have epithelial cells. This is particularly called as retinal pigment epithelium. Okay, so what happens when the light comes, light falls on these cells, then it has a backward movement, and from here, this is the optic nerve, and it goes to the brain. Okay, so here in the retina, there is a photoreceptor layer which comprises of two types of cells. These are the rod cells and the cone cells. Okay, so rods basically function in dim light or night conditions that we say. Okay, and cones functions in bright light or when the light intensity is high. So, and there are specific proteins that are opsin proteins that are present in rods and cones. Okay, so rods have rhodopsin and cones have specific cone opsins for specific colors that determine their type of cone that we are seeing. Okay, so this is a typical structure of the rod and cone cells. So they are also neurons, and this is the cell body. Okay, and because nucleus is there. In case of rods, there is two, both in case of rod and cone, there are two segments, inner and outer. So in inner segments, there is formation of vesicles and the transport of proteins from the cell body to the outer segment. Okay, now these are the outer segments. There are some certain structures called as discs in these region. So why are these discs present here? And specifically opsins that are the proteins that are light capturing proteins are present in these discs. Okay, so for example, if we take an example here, this is one disc, this is second, this is third. Okay, so rhodopsin or any opsin molecule will be present like this on the disc surface. This helps in arranging large number of opsin molecules inside these discs, and this helps in capturing of as much light as possible. And now what happens in case of rods, rhodopsin senses bright light and it stops its signaling. Okay, that's why we are saying that rods sense light intensity. So whenever the light intensity is high, the rhodopsin stops its signaling. And when we talk about cones, so in case of cones, the signaling starts when they are receive some light. So that's why we tell that they function in high light intensity. Okay, so this is the basic functioning of rods and cones, and they how they receive light in our living system. Yes, anyone has any doubt? So if not, then we'll move on to the next question. Yeah. How is cell passaging done? By treating cell lines with PBS, by treating cell lines with trypsin EDTA mixture, by treating cell lines with IMDM, or by scraping the cells. Can anybody guess?
So option B.
Okay. Any more answers?
Option B.
Okay, so the correct answer is B and D. So why I'm explaining it. So let's consider the case of when generally we use poly-L-lysine for coating our plates. Okay, so when poly-L-lysine is used, then in that case, what happens as it's a natural amino acid present in our body systems. So the any other protease, being it trypsin or any other protease, can easily degrade it. Okay, so in our lab also we use poly-L-lysine coated plates. So we use trypsin EDTA mixture. So what we do in this case is like this is a cell that we have to subculture. Okay, it is around 80 to 85% confluent. And in this, what we do, we remove first the media that we have. Okay, for example, we are using complete IMDM, including the serum. First, we remove that, and then we treat with trypsin mixed with EDTA. So EDTA basically chelates all the ions that are present there, and trypsin acts on both the extracellular matrix between the cells as well as the attachment of these cells with the substrate, because these are adherent cells. Okay, so we treat with this, and then we add again the complete IMDM mixture to stop the reaction of trypsin. Okay, so this serum that we have in this mixture basically stops the reaction of trypsin. So we add this again, and then the cells will be detached from the surface as well as from each other. So then we dissolve it in PBS and then we add it in a other plate. Okay, and for like for such cases where poly D lysine is used, okay, in that case trypsin might not work because it doesn't have that capacity to degrade poly D lysine. Okay, so in that case, what we do, we manually scrape the cells. So what we do is we generally have the media in the culture plate. So we wash the cells many times with some force of the pipette to just scrape the cells out of this particular attachment zone. So what that do? That basically detaches the cells from the surface as well as from the extracellular matrix. So it is a mechanical method of scraping the cells. Okay, and this is the enzymatic method that we use in our daily lab practices. So anybody has any doubt?
At what point you said the trypsin will not have any effect?
Sorry, can you repeat?
At what point you said the trypsin may not have an effect?
For the scraping of cells.
Yes, you said
Okay.
the trypsin may not have an effect at some point.
Okay, so generally we don't do scraping. We generally use poly-L-lysine only, but in some cases where the trypsin cannot be used. For example, there is some cases where poly-D-lysine is used as a substrate for attachment of cells. So in that case, we usually use scraping so that we generally mechanically use a scraper that we have. It's a plastic tube with a specific plastic like lifted part that helps in detaching the cells. So we rub this plate with the scraper and detaches the cell from the surface. Or else we can directly wash with the media that it has with some force. Okay. Although there is some what we say loss of cells in this case because there is mechanical degeneration of the cells. But this is also one of the methods that people use for detaching the cells.
Clear, sir. Clear. Thank you.
Okay. Any more doubts? not, then we'll move on to the next question. So we have two surfaces. Okay. In one of the surface, protein is sprayed on the top of a glass surface in a manner that it does not fully cover the original surface. Okay. So considering this as the surface A, we added a protein and it covers only some part of the surface only, and the other part is left. Okay, this is not covered. This part is not covered with the protein. And in the second surface, we have a protein layer coated on a glass in a manner that the original surface is fully covered. So in this case, what happens? The whole surface is covered with the protein. Okay, there is no space left behind. So if we have an XPS spectrum, okay, so X-ray photon spectroscopy we are using here to determine what will be the surface characteristics. So what will be not true from the given options. So can anybody guess? So like XPS basically tells us that it's a chemical characteristics of the surface that we are using. Okay. So considering surface A, we know that this part of the surface is covered with the protein. Okay, and this part is empty. So glass is basically silica. Okay, so it's form of SiO2. So and the protein that we have is the protruding groups will be basically nitrogen. So if we have any protein, it will give some peaks in nitrogen, carbon, hydrogen like this. Okay. So if we see the spectrum of surface A, what will happen? We will have some peaks of oxygen, silicon, and nitrogen. Okay. This is the particular peak pattern how we get in case of an XPS. Okay. And if we talk about surface B, because it is fully covered with the protein, okay, so it will have no space exposed with silica. So if we see the spectrum of this particular surface, this will be like N, O, H, C, like these elements will be present that are particularly part of the protein. So the spectrum of surface B will not show any peak related to silica. Okay, so silica will not be present in that particular spectrum. So if we see the options right now, surface A will show nitrogen and silica peaks. That's correct. Okay, it will show both type of peaks because the surface is partly covered with the protein and partly is exposed. Surface B will also show nitrogen and silica. No, it will not show silica peaks. It will show nitrogen peaks. Okay. Spectrum A and B both will show nitrogen peaks. Yes, both will show nitrogen peaks. And spectrum for surface A has a higher oxygen peak as compared to B? Yes, because here the spectrum, the surface is particularly exposed. So here the oxygen peaks will be more as compared to the protein cover. Okay, so the correct answer would be option B. So anybody has any doubt in this question?
Sorry, we have not understood correctly. Will you be able to repeat it? We couldn't understand.
Okay, so like in what does an XPS spectrum tells us? It tells us the chemical characteristics of the surface. Okay, so whenever we take a glass slide, suppose it's a surface but that is not exposed to anything, that is not covered with anything. It is only a glass surface. So whenever we will do a XPS spectrum for this, so what we will get? We'll get such peaks. Okay, and that will be mostly of silica and oxygen. Okay, this is for only the glass surface. Whenever we'll cover it with some protein. Okay, so first take an example that only half of the slide is covered with the protein. Okay, so now if we get a XPS spectrum of this, what kind of peaks can be, what kind of elements can be visible from the, like can be detected from this thing. So we might get peak from silica, oxygen, nitrogen, carbon, and many more elements that are present in proteins. Okay, so here we are only talking about the number of peaks that we get with the number of elements present on the surface here. That will be detected by the XPS. Okay, now the question says that in this case, where there is the surface is partly covered with the protein, so in that case, what additional peaks can be visible? So as the glass surface is also visible, we'll get some peaks of silica and oxygen. Okay, but as protein is present, so we will get some peaks of nitrogen, carbon, oxygen like this, and so on. But if the protein covers whole of the surface of this glass slide, that means silica or the glass surface is not open. Okay, for the XPS spectrum to analyze its elements. So the elements of the glass are in contact with the protein. So they are not open for the analysis. Okay, so in that case, what we'll get peak that will not have silica in that because oxygen can be a part of the protein itself. So we'll have peaks from oxygen, nitrogen, carbon, and so on, the elements present in the proteins. Okay, this is how we get these peak spectrum of from XPS instrument basically. So this helps in determining what all elements that we have on the surface.
Thank
Okay.
you, sir. Clear.
Okay. Any more doubts? If not, then we'll move on to the next question. So poly-D-lysine is a natural or synthetic polymer that can bind to carbohydrates or not bind to carbohydrates. Can anybody guess?
Option A.
Okay. Any more answers?
Option D, sir.
Okay. So yes, the correct answer
Option
is option A. So poly-D-lysine is basically a synthetic polymer, and that binds to the other cells or other proteins based on the electrostatic interactions. So it doesn't generally bind to carbohydrates directly. That's why if we are using any carbohydrate related thing, we'll use other substrates for attaching the cells. Okay, so it doesn't bind to carbohydrate. Okay, so let's move on to the next question. So out of these four cell lines, which one is a neural cell line? Can anybody guess?
D, sir. Option D.
Okay, any more answers?
D, sir. Option D.
Okay, so I hope everybody knows what type of cell line HeLa is. It's a
It's a cervical
yes,
cancer, HeLa.
yeah, it's a cervical cancer cell line. Okay, and then we have NCLH295R. This is basically an adrenal gland cell line, adrenal cortex cell line. Okay, and then we have CaCO2. This is epithelial cell line that is derived from uh, the intestine of the digestive system. And then we have NG10815. This is a hybrid cell line of glioblastoma from mouse, and glioma from rat. Okay, so this is the neural cell line that we use in our cell culture system. Okay, so the correct option is answer D. Okay, so let's move on to the next question. So rods and cones are electrically active, sound sensors, pressure sensors, or electrically inactive. What can be the correct answer? Can anybody guess?
Option A.
Okay, any more answers?
Option B, sir.
Option B. Okay, any more answers?
Option A. Option A.
Uh,
Electrically active. Option A.
okay. So as we talked before also, like in this case, yeah. So when light falls on this particular rods and cones, what happens? Signal transfers from these cones to the optic nerve. Okay, following various layers of the retina. So this particularly signal comes from the electrical activity of the neuron cells. Okay, so that's why they are called as electrically active. Okay, so rods and cones are basically electrically active neuron cells that perceive light and that sends the signal to the brain that, okay, this thing we are seeing. Okay, so the correct answer is option A, they are electrically active. Okay, so let's move on to the next question. So to prevent detachment of collagen from plastic substrate, it was that fill in the blanks to the plastic using carbodamide. So why we are using it? Can anybody guess? So what happens, like on glass surface also, we might have some cross-linking agent. So if we have a plastic surface, what happens? Sometimes the collagen falls off from this plastic surface. So to prevent that detachment, what we do is we have a layer of carbonamide that has some functional groups. Okay, that has some active functional groups that helps in cross-linking to the collagen molecules to prevent its detachment from the surface. Okay, so it is basically cross-linked via bond formation to help the collagen molecules to attach to the surface, and with time it does not detaches from the surface. Okay, so option B is correct here. Okay, so anybody has any doubt in this question?
Sir, then at the time of passaging of the cells, we face problem.
Yeah, for that case, what we'll do is we'll have some other reagents also to like detach the cells from the surface, as well as we can use the mechanical method of scraping also in that case. Any more doubts? If not, then we'll move on to the next question. Which of the following techniques can be used to understand the depth profile of the surface of the substrate? So can anybody guess?
Option A.
Okay. Any more answers?
Option C, sir.
Okay. Any more answers?
Option B, sir.
Option B. Okay. Any more answers? So, like, from the contact angle measurements, what we see, we generally get the hydrophobic or hydrophilic profile of the surface. Okay. So this cannot give us the depth profile of the surface. That what are the molecules present on the surface and how much deep they are present. Okay. X-ray photoelectron spectroscopy will give us the chemical analysis of the surface. Right? And same with the paramagnetic resonance spectroscopy. It basically tells us which type of element is present there. Okay. Now, if we have to see, for example, this is the substrate. Okay. This is the glass slide that we use. And above this, we are coating it with the substrate. And it will form like these ridges. Okay. So how do we know what is the surface characteristics of this particular glass surface that we are using? So for that, we use atomic force microscopy. Okay. So how do we use this? The basic principle is explained in this figure itself. So it has a probe. Okay. So as you can see, the probe is like this. it's a needle that has a small cantilever. This is called as a cantilever. So what will happen? This is the surface that we are studying, and what it will do is it moves in a back and forth direction. So it goes touches the surface. It will not directly touch the surface. It is like when there will be van der Waals interaction between the two molecules that are present, one on the surface and one in the cantilever. So it that just directly goes here and comes back. Okay. So first it will go have like see what is there and then it will come back. Okay. Now with this probe, we have a laser like in this instrument, we have a laser attached. So when this probe will go to the surface, the laser will be put on this probe. That will be reflected and at like there it will be detected by a photo detector. So what will happen if the surface is has a depth? So the cantilever will go here and it will like have a different angle of reflection. And if the surface is has a hill type of a structure, so the cantilever will only be like here. And what will happen? Then the reflection angle will be different as compared to this one. So from this, a particular image of the surface will be formed. Okay, so the like the surface is like this, like this. So this is how the atomic force microscopy works. So the cantilever will go first here, then the laser will be sent and the reflection angle will be measured. Then the cantilever will move forward and will go here, here, here. Like that, we'll measure the depth of each and every molecule present on the surface. Like this, it will form an image and that will tell us the depth profile of the surface. Okay. So anybody has any doubts in this particular question or anything which is not explained, can ask.
Sir,
Hmm.
AFM can tell us the porosity of immobilized cell.
It cannot tell the porosity basically because like porosity tells us the space that is vacant after we added any substrate on the surface. Okay. So it will not be able to tell the pore size or anything like that. It will only tell, okay, so here the pore is present, but it will not be able to tell the pore size or the volume that we have for the cells.
Okay, sir.
Any more doubts in this particular question? So if not, then we'll move on to the next question. So yes, a person stained culture flask with poly D lysine. He used 1 mg per ml solution of poly D lysine and cultured the cells in the same flask after coating the surface. Okay. So he found that the cells did not grow properly. What can be reason for the same? The concentration of poly D lysine was the used was more, or it was less, or some other reagent was used as compared to poly D lysine, or none of the above.
Option A, sir.
Option A, sir.
Okay. Any more answers? So yes, the correct answer is option A. And the reason is we know that poly D lysine that is used in case of the coating purposes is used only like less than 100 microgram per ml. Okay, this is the threshold that we have for coating the cells for cells growth. Okay, so if we use anything greater than this, that decreases the growth of the cells. Okay, so this is the reason if we are using more than here we have used 1 mg per ml solution of poly D lysine. So here the cells will detach from the surface. Okay, and they will not grow properly. So this is the reason for it. Okay, so let's move on to the next question. So here we have some values from the XPS spectrum. Okay, so as we know, XPS is used for chemical analysis of the surface. And it can be both qualitative and quantitative. Qualitative in the sense it will tell us what elements are present there, and quantitative in the sense it will tell us how much amount of those elements will be present there on the surface. So we have this particular values obtained from two surfaces A and B, and in both the surfaces oxygen, nitrogen, carbon, and silicon is present. So what can you deduce from these particular values that we have? So link this particular question with the one that I discussed previously and then answer.
Option B.
Option B. Okay. Any more answers? So like we consider first the surface A. Okay, so this is the surface A, and considering the fact that it is not coated with anything or any protein or any polyol lysine, so it will what it will the spectrum XP spectrum will give, it will give oxygen, silicon, and some more metal. If it is partially covered or some part of this is covered with the polyol lysine, then it will give some peaks with this. Okay. Now, if this surface is fully covered with polyol lysine, that is properly there, no surface is being exposed. Okay. So in that case, it will not give any peak with silica. It will only give peaks with this. Okay. So the correct answer will be surface B, that it is properly coated with polyol lysine because it is not giving any peaks with silica. Okay. So the correct option is option B. So anybody has any doubt in this case? So if not, then we'll move on to the next question. So this is a calculation based question. So you have a 15 centimeter square cell culture plate that is round in the structure like this. We have the cell culture plate. So you are coating it with collagen. Okay. You have used 10 microgram per ml of the solution, collagen solution, and out of that 3 ml you have added in the culture plate to collect it, like to coat it. And you have nothing thrown from the solution after adding it to the plate. So everything was absorbed on the surface and nothing was removed after that. Like after that, a thick gel was prepared out of that. That was uh, 2 micrometer. This will be 2 micrometer. Okay. And was formed on the surface. What will be the total volume of space that will be left unused by collagen, considering that the whole plate is coated with collagen? So you have molecular weight of collagen, length, and diameter. Okay. So try to solve this question. We'll discuss in few minutes. So basically, we have to calculate the pore size, pore volume that is made after collagen uh, coating on the surface. Okay. So can anybody tell the process? How will we calculate this? So in this case, we have a culture plate. That surface area is 15 centimeters square. Okay. And in this case, we added collagen, and that the concentration was 10 microgram per ml. And out of that, 3 ml was added. Okay. Now, it formed a 2 micrometer thick film on the surface. Okay. So what we have to calculate is, after the full surface was covered with collagen, there will be some pores left or some pores formed after the coating with collagen. So we have to calculate the volume of the pores that we have. Okay. So how will we calculate it? First, we'll determine what is the mass of collagen that went inside the plate. Okay, that will be 10 into 3, that is 30 microgram. Okay, and this 30 microgram will have how many molecules of collagen? Okay, so how will we calculate the molecules of collagen? For that, we have the molecular weight of collagen, that is 33 kilodalton. And for Dalton to gram conversion, we can use directly. So 1 Dalton is equal to 1 gram. So here the molecular weight will be 300 kilogram. This is for approximation purposes only. Okay. So and then we need the Avogadro number. Okay, so Avogadro number is 6.02 into 10 to the power 23. So from this Na and the molecular weight, we'll calculate the number of molecules present in 30 microgram of collagen that went inside the plate. Okay, now we have the number of molecules present in collagen. Okay, now we have the diameter of one collagen molecule and the length of one collagen molecule. These are the dimension of one collagen molecule that we have. So consider collagen as a cylindrical structure. So its length is 280 nanometer and its diameter is 1.4 nanometer. Okay, so now for one collagen molecule, the volume will be volume of the cylinder. Okay, so that will be pi r square h. So from this, we'll calculate the volume that will be covered by these much molecules of collagen present in 30 microgram of solution or 30 microgram. Okay, that will give us the volume covered by the collagen on the surface. Now, this collagen molecule formed 2 micrometer thick gel. Okay, and that too on a surface of 15 centimeters square. So the total volume covered by this collagen will be 15 into 2 micrometer. Okay, this will give us the total volume of the plate that is covered by collagen solution. So now what we'll do is we have the volume of the total collagen molecules present in that particular solution, and we have the volume that is filled, the culture plate is filled with the collagen solution. So now, in order to find the space that is vacant after like the solution is poured into the culture plate, we'll subtract the volume occupied by the collagen molecule from this particular volume. So that will give us the space that is left behind in the culture plate. So now, can you guys calculate? Or shall I explain it again?
Sir, could you please explain again?
Okay. second. Just a second. So what we have in this case, we have a plate that is 15 centimeter square in area, that is surface area. Okay. Now, what we have done is we have to code the surface area and we have added 10 microgram per ml of collagen solution and that to 3 ml. Okay. So after adding that, what we have is we got a gel kind of structure that is 6 micrometer thick. Oh, sorry, 2 micrometer thick. Okay. Now we have to calculate after the full solution of collagen is being coated on the surface, there will be some pores created in the gel. What will be the pore volume or the volume that is empty after coating of collagen inside the plate? Okay, so that will give us the total pore volume of the whole plate after coating with collagen. Okay, so now what we'll do is the first step. How will we calculate the collagen volume that is being occupied by the collagen on the surface of the plate? Okay, so for that, what we need is we have to have a volume of one collagen molecule. And secondly, we must have the number of molecules of collagen put on the plate. Okay, so first, volume of collagen molecule can be calculated by these dimensions and consider collagen as a cylinder molecule. Okay, so it has a length of 280 nanometer and a diameter of 1.4 nanometer. So the volume of this particular collagen molecule will be reduced by the formula pi r square h. Okay, so this solves us with the first problem that we have. Fine. Now, how to find the number of collagen molecules that we have in particular in this particular surface. So we added 3 ml of 10 microgram per ml of the solution. So how much amount of collagen will be added in this particular surface? So that will be 3 into 10 microgram per ml, that is 30 microgram. This much amount of collagen we put in this particular cell plate. Okay, now from this, we know for calculating the number of molecules, we need two more things. That is the molecular weight of collagen, that is 300 kilodalton, and the Avogadro number, that is 6.02 into 10 to the power 23. Okay, so here we apply the mole concept. Now, as we know that number of molecules present in the molecular weight of the molecule or the number of molecular weight of the molecule is Na times. Okay, so in 300 kilogram of collagen molecules, we have 6 into 6.02 into 10 to the power 23 molecules of collagen. So how much molecules will be there in 30 microgram of collagen that we have to calculate? Okay, so this will give us our second problem, that is the number of collagen molecules that we have. Now, after this, we'll calculate the total volume of these much molecules of collagen that we have on the surface. Okay, So how will we get that? We'll get that with the total volume Vt we will be noting. So this will be get by volume of one collagen molecule into the number of molecules of collagen. Okay, this will give us the total volume of collagen occupied on this particular surface area. Fine. So till this part, is there any doubt?
It's clear, sir.
Okay. Now, what we'll do is we have to calculate the empty space now. So what we'll do is we know that 2 micrometer thick gel was formed and the surface was 15 centimeters long. So what will be the volume of that particular uh, gel? That will be 15 into 2 micrometer. Okay, so that will give us the volume of the total collagen solution covered on the cell plate. Now, to calculate the empty space, what we'll do is so volume of empty space will be this volume minus Vt. Okay, so this will give us the volume of empty space, or this will give us the volume of pores. So am I clear?
Yes, sir.
Anybody has any doubt in this particular case? So the answer that will be coming, that will be near to 2.97 millimeter cube. This will be the volume that will be empty inside in this case. Okay. So if anybody doesn't understand this, can like go have a look on this problem. We can discuss this in the next session also. Okay, so let's move on to the next question. So you are studying drug release kinetics on HeLa cells. So you require slow drug release to the cells. What must be the porosity of the substrate for the same? Can anybody guess?
Option B.
And B. Okay. Any more answers?
Yes, sir.
A. Okay. Any more answers? So always remember, like what we consider is whenever there is high concentration of anything, okay, it will have a low pore size. Okay, and for slow drug release, we'll require low pore size every time. So, for example, here we have a cell culture plate, and here we are growing the cells. And if the pores are smaller, it will like resist the drug molecules to release very frequently to the cells. Okay, if the pore size will be larger, what will happen? The drugs will be easily accessible to the cells that we are treating. So in this case, we have low pore size for slow drug release. Whenever we need a slow drug release, then we'll have a low pore size. And for obtaining low pore size, what we do is we'll have more concentration of the substrate that we are coating the slide with. Okay, so this is the basic concept that we use in this case. And if we have a fast drug release, in that case, we'll use a high porosity. Okay, so the correct answer is option A. So anybody has any doubt in this question? If not, then we'll move on to the next question. So what is the role of nitric acid in coating glass cover slips? It helps in etching of glass, helps in removal of grease, helps in making the surface rough, all the of the above. So can anybody guess?
D, sir.
Okay, any more answers?
Option D.
Okay, so yes, the correct answer is option D. And what we generally do is, like in case of cover slips or glass slides or any other glass surface that we get from market. So what we do is first we wash, for example, this is the cover. So wash it with soap solution. Okay, then we use any acidic thing to etch the glass or to remove the grease from the glass surface. So we can use concentrated hydrochloric acid or nitric acid, or some people use hydrogen bromide also. Okay, to make this glass grease free and to make the surface rough so that easy coating can be done out of these particular glass surfaces. Okay. And why is this needed? So that the coating will be more perfect in this case. Okay. If there is any grease or any other thing that is left on these particular surfaces, then the coating will not be perfect and that will hamper our experiments. Okay. So this is the thing that we use in like one of the process that we use in coating of the surface. Okay. Then after using any of these acidic components, we wash the slides so that any acidic component is not remaining on the slide surface. And then what we do is we generally uh, store it in alcohol and then we coat the surface with the particular substrate that we use. Okay. So this is the basic procedure that everyone follows in their life. So anybody has any doubt? If not, then we'll move on to the next question. So photoreceptors are isolated from that. So what is the preferred substrate that we use for photoreceptors? Can anybody guess?
Option B.
Okay. Any more answers? So yes, the correct answer is option B. And this is the particular uh, structure that is visualized by treating the cells with the concavalent aid. So what happens is, like this is the rod cells that we see, and this is the whole outer segment present inside the rod. So I explained that it has this surface that basically increases the surface area for rhodopsin signaling. So that's why this is the long outer segments that we see. And in case of cones also, we have a shorter segment, but this is also used to increase the surface area for light intensity adaptation. Now, why we use concavlin is specifically for photoreceptors because in case of like concavlin A is also a lectin molecule. Okay, that is carbohydrate binding proteins that we have. So, uh, how this helps is in case of photoreceptors, the outer segment structure is cell membrane is full of carbohydrates. Okay, so this lectin will help these particular photoreceptors to adhere to the surface. Okay, but if we use any other substrate, for example, poly-L-lysine or poly-D-lysine, in that case, it will not be able to adhere these photoreceptors because the cell membrane is carbohydrate rich. Okay, so this is the particular reason why we use specifically concavulin A for photoreceptors. Okay, so anybody has any doubt in this question?
நீங்க எப்போ பணம் எடுத்தீங்க 10 ரூபாய் எடுத்தீங்களா எடுத்தது காட்டுது 23/8 எடுத்தீங்களா 9500
So anybody has any doubt in this question? If not, then we'll move on to the next question. So laminins are used for which type of cell culture? Or typical cell types that we use lamins for? Can anybody guess?
Option C, sir.
Okay, any more answers? So laminins are one of the natural substrate that we see. Okay, so these are in our body. These are generally present in those cells that are basically helps in differentiation. Okay, so all the cells that are differentiating, basically stem cells or any other cells that gets differentiated into some other particular cells. So these laminins are present there. And in cell culture system, these are also used for specifically differentiating cells. Okay, so these are specifically used for embryonic stem cells that helps in differentiation. Okay, so option B is the correct answer. Laminins are specifically used for differentiation or to study the differentiation in specific cell types. So embryonic stem cells usually we use them for differentiation purposes. So that's why laminins can be used for culturing embryonic stem cells. Okay, so let's move on to the next question. So poly-L-ornithine is used in cell culture for. Can anybody guess?
Option B,
Option
sir.
D.
Okay. Any more answers?
Option D.
Okay. Any more answers? So ornithine is also a positively charged amino acid. Okay, but it is not present that frequently as compared to lysine in our body. So ornithine can also be a substitute for poly-L-lysine that we use, or poly-D-lysine that we use. It's a substitute for this particular compound. So it can adhere to any set type because of its positive charge. Okay. And in specific cases, it is used for attaching lamins to the surface because lamins don't directly bind to the glass surface. So we want to have a first coating of ornithine followed by lamins to get it adhered to the glass surface. And then similar to laminins, fibronectin also like have problem in binding with the substrate directly. So they also require a particular second coating of any other compound to get it to get adhered to the surface. So fibronectin is also used for ornithine is also used for attaching fibronectin to the surface. So all of the above is the correct answer. Okay. So fibronectin along with ornithine is used to grow neural cells. Okay. So this combination is basically used for growing various neural cells for their attachment. So the correct answer is option D. Okay. So this marks the end of our session for today. So if you guys have any doubt in this presentation or from any topic from the last week, can discuss.
There was a question early in the discussion. Hemoglobin is a glycoprotein for application. You mentioned about influence and all. Can you go back to that question?
Okay. This one.
Yes, correct. This one, sir.
So actually what happens is hemagglutinin is present generally, for example, if we take an example of influenza virus, so it's present as a recognizing molecule. So we recognize influenza viruses as H and N series. Okay. So in that H stands for hemagglutinin. Okay. So it's also a lecithin molecule that is a protein that binds to carbohydrates. Okay. So what happens in this case is whenever we have took the example of influenza virus only. So in case of an influenza virus, this is a glycoprotein, surface glycoprotein basically. So it binds to the RBCs that we have. So RBCs have these blood groups like ABC, sorry, A and ABO blood group system. So in that case also, the blood groups that we have has modified carbohydrate moiety on its surface. Okay. So this particular hemagglutinin basically recognizes this carbohydrate moiety and binds to the cells. And inside, when infection happens, so what happens is these virus molecules basically agglutinates the RBCs. So the process is called as agglutination in that case.
The correct answer is D, glycoprotein agglutination.
Sorry, can you please repeat?
So the correct answer is D, glycoprotein agglutination.
Yeah, the correct answer is option D.
Thank you, sir. Clear.
Any more doubts? So if not, then we close the session for today. Thank you guys for your patience.
Thank you, sir.